Assalamualaikum warahmatullahi wabarakatuh. Hi, class. Alhamdulillah. Now we have learned on fertilization for chapter 7. So I want to recall back what we have learned before. So here you go. Okay. Let us recall back all those how the sperm will become in contact with the ovum itself. So it's a lot of layers for the ovum, from corona radiata, zona pellucida, and all of those should be go through by the sperms, only one sperm. So there are a lot of reactions that occur from acrosomal reaction, the binding of the sperms towards the ovum itself, the oocyte itself, the fusion what are occurring, and the block of polyspermy, what we call as after fusion or cortical reaction. For the block to polyspermy, maybe it can be fast block or slow block. Okay, this is two terms that we have learned before. Okay, so now we want to continue on how the ovum will complete the second meiotic division after sperm entry, and then after that will undergo mitosis for the development of the zygote and so on. So there are phases of fertilization that include the fusion of oocyte and sperm. So the fusion of the membrane plasma of both oocytes and sperms are what we consider as the fertilization. As the head and tail of sperm enter the cytoplasm, but the sperm plasma will remain behind, and the secondary oocyte are actually arrested in metaphase second of meiotic division. Now it can continue after fertilization occur into anaphase and telophase. So now they are forming a mature ovum. So this mature ovum can fuse with the sperms to make a pronuclei, what we call as formation of male and female pronuclei. Means the nucleus itself are now ready to be combined together. So what we call as the pronuclei. Pronuclei is when the chromosomal material from the sperms, okay, for the sperm itself and from the ovum itself, both can be condensated and become enlarged. Means the nucleus itself are becoming matured. And following the completion for ovum, it's not only decondensates and enlarge, but also can now complete the meiosis too. Means all the cycles now are there. At this stage, the female and male pronuclei, both of them cannot be distinguished. Which one is from male, which one is from female. So so we cannot distinguish anything. But this one is just form of illustration. As they grow, the pronuclei replicate the DNA, and the pronuclei itself is only in form of sperms and ovum. Means they are only haploid. Okay, then after this fusion, what are occurring? The pronuclei formation. We can see the pronuclei formation. The sperms will lose the flagellum, and this only remain for the nucleus, what we call as male pronucleus. And the ovum itself extrudes all its second polar body, and the remaining is only female pronucleus. And now the pronuclei formation, when they are united together, what we call as syngamy, the process for combination between pronucleus of male and female, and it is called as a true fertilization. And the pronuclei from the haploid nucleus of a sperm or an egg before fusion of the nuclei in fertilization, now it can become a second one. Okay, the pronuclei formation after that, two chromosomes of the groups means groups from where? Groups from male and groups from the female. They are now united to form a single group. So these are representing a process of the first cleavage mitosis. Means now it's not occurring meiosis, but it's starting from mitosis of, okay, now they are starting to undergo mitosis of prophase. Okay, then fertilization now it can considered as complete. And then once it is fertilized at the part of the uterus, now it can be implanted and referred as conception. So you can see pronuclei formation after cortical reaction. So you can see the male pronucleus and female pronucleus. Now they are what we call as decondensated and enlarges, both of them. And now for some phases, the syngamy are occur, means the pronucleus both are as well as the nucleus fused together, and this is what we call as only one single group. And now they are completing its chromosome in 46 numbers chromosomes. So what are the results of fertilization? First one, it stimulates secondary oocyte to complete meiosis. From metaphase only, it now can directly go to anaphase and telophase. It can restore normal diploid number of chromosome. Okay, means they are undergo mitosis then, and results in variation of human species and the embryo that contain only maternal mitochondria, because of the sperm mitochondria at the neck of the mitochondria itself has been dissociated and then discarded and dispersed into the egg cytoplasm. That's what we call as the mitochondria DNA. It's only from the mother itself. And then from fertilization, it can determine the sex of the embryo, whether because of the sex chromosome that carried by the father. If the sex chromosome of the father are carrying Y, means the mother will carry X. So now the baby will become a boy. If the sex chromosome is carried by the father is X, so both are X, means the sex of the embryo will become a girl. So how's about the twin? Okay, twin can be second types. There's two types of twin. First one, monozygotic. Okay, means only a single cell of eggs will split into two developing zygotes. So these are occurring at a very early stage, means it's fused, it doesn't fuse, and it comes into, it does not fuse, it splits into two. This is what we call monozygotic, or what we call as identical twins. But if it is dizygotic, it's from two sperm and fertilized two separate ovum. So because by hook or by crook, both of them a fourth of them reach the maturation at the same time. Means from a normal pregnancy, it's only one sperm and one ovum. But for dizygotic, there's two sperms and they are two separate ovary. No, sorry, they are two separate ovum. So the twins are not identical because of this dizygotic, and they are having mostly different sexes, and this one can increase with the age of mother. So for the summary of fertilization, fertilization is the passage of sperm through the corona radiata, and then it penetrates the zona pellucida, and all those enzymes that are involving from esterase, atkosin, and neuraminidase. And then the fusion of the oocyte and sperms following there, what we call as the fusion then from this fusion, the oocyte complete the cyclomatic division and it form a female pronucleus, as well as the sperms also form a male pronucleus. And then now there are cortical reactions, means the fast and slow block of polyspermy, fast and slow block of polyspermy. Then now it's formation of pronucleus. Then after the breakdown of pronuclear membrane, the condensation of the chromosome and the arrangement of chromosome for mitotic cell division. So basically there are 7 steps there for fertilization. So this is an illustration from the first type of the sperms that are going into to fertilize one single ovum. So there are hundreds of sperms that attracted to the corona radiata, and they are trying to break down the barrier of the cells granulosa. Then once it becomes in contact, only one of the sperms become in contact, it triggers a chromosome reaction. So it secretes the digestive enzyme that breaks down the glycoprotein of the zona pellucida and helps to expose the oocyte's plasma membrane. And the single sperm succeed in burrowing, means it's like trying hard to enter the ovum. So once it's become in contact with oocyte plasma membrane, and the sperm's plasma membrane itself will fuse to the plasma membrane. Okay, this plasma membrane of the sperms will fuse to the plasma membrane of the oocyte, and sperms release its nucleus into the cytoplasm. Only the nucleus, all of those neck and tail and so on will be dispersed out from the eggs. So what we call as how's the infertility can be, or fertilization go away, how it can happen. Okay, if there's too many sperm, what we call as dispermy or triploidy, it can lead to abortion. Means it spontaneously can trigger abortion in most of the cases. And fertilization also cannot be if there's infertility cases, means there's a lot of benchmark in order to know whether they are fertile or not. And then bad timing also can lead to fertilization go away if the sperm and the ovum are not meet together at the same time, so they cannot meet. So what we have discussed before, the sperms and only can survive for 48 hours, but for the oocyte or the ovum can be there only for 24 hours. So if the bad timing occur, so there's no fertilization occur. So now we want to discuss about polyspermy. Polyspermy means supernumerary sperms. Extra sperm can successfully enter the vitellus of the zona reaction. So how it's occur? It's only 2% of the mammals that are occur, and usually it's because of the ovum that's huge in number. And if there's delay in copulation, it can increase the condition. For example, in pigs, and polyspermy can lead to polyploidy. Polyploidy can, okay, this one like triploidy, right? It's only should be 2 nucleus, but this one is 3 nucleus. So it can cause lethal, means the mutation are occur, and this embryo usually will be discarded. If not, if we cannot trace it earlier, if it is fertilized or developed inside the wombs, it will always abort spontaneously. So these are differences between normal cleavage and polyspermy. If there's polyspermy, there is a lot of it's not firm, and there's a lot of spaces between all those cleavage. And then timing of mating. For the fertilization, if we are discussing about the cows itself, it can be in the best condition. Is it inseminated from the middle to late estrus from 12 to 18 hours? And why it should in the middle? Because there are time for capacitation occur for the transport of the sperm and to get a fresh male and female gametes. Okay, then what we have considered about fertilization is on the fertility, right? So the fertility means kesuburan orang tu. So we do have the analysis. There's 7 parameters that describe whether the semen are healthy or not, based on its volume. Liquefaction time means the volume itself, how much semen is present in one ejaculation. For the liquefaction means the semen itself is a thick gel, but it can becomes a liquid. How? After 20 minutes after evacuation. So liquefaction takes time to turn into liquid, so that from this liquid means it can easily move, right? So means it's also can affects the healthy or fertility of someone. Okay, then we do have sperm count. How many sperm present per milliliter for semen in one ejaculation? Then sperm morphology, measure the percentage of sperm that are having normal in shape. For the sperm motility, it measure the sperm percentage that can move forward normally. So how it's move forward, and amount of semen also can be measured. It's called as motility density. Then we do have semen analysis in terms of the pH, how the acidity as well as how's the alkalinity of the semen itself, because it should be in optimum condition whereby it can pass the cervix of a woman. Then white blood cells count also not normally present in semen. If it is present, means it's, if white blood cells is there in semen, means the semen is low in quality. Then fructose level is the measurement of the sugar, and actually for the fructose level is providing the energy for the sperms to swim. So these are the references values for semen analysis. For the volume, it should be for one ejaculation more than 2 mils. The count more than 20 million of of sperms in 1 meal, and the motility should be more than 50% progressive, and if it is progressive, and it should be 25% of it should rapidly progressive, and the morphology should be more than 15% normal, and white blood cells should be less than 1 per mil. So these are the terms for semen analysis. If the semen analysis normozoospermia, means it is normally ejaculated. If the name is aspermia, means there's not ejaculating at all. If it is zoospermia, it is no sperm in ejaculate. They ejaculate, but there's nothing. There's nothing inside, means there's no sperm itself. And then we go to the sperm concentration. If the sperm concentration less than 20 million per mil, it's called as oligozoospermia. If there's movement abnormality, we call as stenozoospermia. If the sperms less 30% of it has normal morphology, means the morphology are obstructed, it's called as teratozospermia. If the combination between these three, okay, it's considered as the term of oligoastenoteratozospermia, means disturbance of all of these three variables. So these are all those terminologies for semen analysis, okay. As what we have discussed before, they do has aspermia, no semen volume at all, pyospermia or hematospermia if there's red blood cells present in the semen. Pyospermia is the leukocyte present in the semen, or necrozoospermia if there's dead sperm inside the semen. And then in terms of the volume, should be if the volume is lesser than 1.5 mil, it's called as hypospermia. If the volume semen is more than 6 mil, so it's called as hyperspermia. Or azoospermia if there's no spermatozoa in the semen itself. And then we can do macroscopic examination, means a gross, while we can see by our naked eyes only. So the macroscopic examination can give a description. For example, how's the appearance? What are the normal? Okay, the normal appearance should be whitish to gray opalescence. Okay, but if it is appear in yellow in color, means there's something urine or jaundice. Or if it's the color pink or reddish or brown in color, means there's red blood cell inside the semen. Okay, then we do have liquefaction. Liquefaction is, it should be only 20 minutes to turn from the gel into liquid. So if it is lumpy, more than 60 minutes, the sperm also can be trapped. Okay, so why? Because this one is the sign for prostatic infection or lack of prostatic protease. So the enzymes is lack there. The viscosity should be smooth and watery, but it is abnormal if it is thick with long threads like 25 G needle. So the high viscosity can impede, means slower down the sperm's movement. Then you can see in terms of the volume itself, it should be 1.5 to 2 mil per ejaculation. If it's lack of the volume, it represents a block or retrograde ejaculation, infection, and lack of androgen. And because of the low semen volume, cannot neutralize the vagina acidity, and high semen volume can dilute sperms or active infection. Then we do know the normal pH of this of the semen should be more than 7.2 or in alkaline stage, because inside the uterine it is acidic. So acidity less than 7 indicates a congenital, means the man itself from baby has an absence of vas deferens. So vas deferens itself are loose. And the pH increased with time as natural buffering capacity of semen decreased. Therefore, the pH is not clinically useful. So then, it's done for today for the chapter 7. And now, and then I will continue again on chapter 8 for development of embryo. Thank you so much. Assalamualaikum warahmatullahi wabarakatuh.